Proteins are wonderful. They do so many things to help organisms survive, not just helping people build muscle, but also regulating blood glucose levels or forming silk to help spiders catch their prey. But how exactly do they do all that they do? Proteins rely on their structure to function. For the purposes of this video, let's compare them to origami. This paper is folded in this specific way so that it can interlock with identical modules to create this icosahedron structure. And the paper modules in this structure are folded slightly differently to create this different shape. These two have the same base shape, icosahedron, but because of the slight differences in their folds, their structure and thus function are different. Proteins don't always automatically fold into the correct structure. They can misfold, like if I fold the paper in the wrong way, and then this causes it to no longer fit in with identical modules, and it can no longer serve its proper function. But there are ways to prevent misfolding. Proteins can sometimes have chaperones, other proteins that help them fold properly. There are some misfolded proteins, though, that are near impossible to unfold or degrade, such as prions, misfolded proteins that cause other proteins to misfold and eventually lead to death, though they're pretty rare. You can also unfold proteins through something called denaturing, where something like a strong acid or a high temperature can cause the bonds that hold proteins together to break apart. These messed up proteins don't stay in the cell forever, though, and clog it up. An enzyme, which is also a protein, degrades misfolded proteins, so its amino acids can be recycled to make proper proteins. Side note, isn't it amazing and kind of crazy how the solution to mess up proteins is just to add more protein to fix the problem? On the other hand, there are denatured proteins that can return to their original properly folded state. But why? And why do folded proteins stay in their correct configuration? Proteins have specific energy areas that they like to hang out in. Imagine mountains and valleys. Proteins like to chill in valleys in a specific stable structure, and it would take a lot of energy to climb out of a valley to change the protein into another form. But sometimes going into that valley is a lot easier than getting out of it, which is why some denatured proteins will just end up refolding into their proper configuration, and they just fall right into the valley here. This is all due to the free energy of the proteins, and also prions. The reason why they are near impossible to unfold or degrade is because they're in an energy valley so deep it requires a tremendous amount of energy to break the bonds that hold it together and drag it out of its valley, so they just stay misfolded. Proteins are incredibly versatile, just like origami, and they really deserve more credit for all they do for us and for each other.